Students, in this lecture, Inheritance and Variation Part 3, we will study about chromosomal theory of inheritance, chromosomes in that number of chromosomes, structure of chromosome, types of chromosomes, sex chromosomes, linkage and crossing over. First, we will see about the chromosomal theory of inheritance, why Mendel's theory was remained unrecognized. So there are some reasons. First is communication was not easy in those days and his work could not be widely published. Secondly, his concept of genes or factors in Mendel's bird as stable and discrete units that control the expression of traits and of the pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. And finally, he could not provide any physical proof for the existence of factors. Rediscovery of Mendel's result. In 1990, three scientists, De Vries, Porens, and von Scheerbank, independently rediscovered Mendel's result on the inheritance of character. Chromosomal theory of inheritance. This theory was proposed by Walter Sutton and Theodore Boveri in 1902. They worked out the chromosome movement during meiosis. The behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's laws. Sutton united the knowledge of chromosomal segregation with Mendel's principle and called it the chromosomal theory of inheritance. Chromosomes and genes are present in pairs in diploid cells. Homologous chromosomes, homo means similar, separates during gamete formation, that is the meiosis stage. Fertilization restores the chromosome number to diploid condition. The chromosomal theory of inheritance claims that it is the chromosomes that segregate and assort independently. Chromosomes. What is a chromosomes? What are the chromosomes? The chromosomes are filamentous bodies present in the eukaryotic nucleus. The term chromosome, it is a chroma means color, soma means body, and this term was coined by W. Waldeyer in 1888. The size of chromosome varies from species to species. Chromosomes are visible during cell division. They are capable of self-replication and play a vital role in heredity, mutation, variation, and evolutionary development of eukaryotic chromosomes are made up of DNA, histone, and non-histone proteins. Its function, its chromosomes are carriers of heredity. Next, that is the number of chromosomes. The number of chromosomes is specific and constant for a particular species. Therefore, it is of great importance in the study of phylogeny and taxonomy of the species. So, this is the term ploidy. The ploidy speaks for the degree of repetition of the primary basic number of chromosome, that is X, in a cell. When chromosome number in a cell is exact multiple of the primary basic number, it is called as a euploid. Euploid includes monoploid or haploid with one set of chromosome, that is X or N. X is equal to N. Diploid, that is 2N, two sets of chromosome. Triploid, that is three sets of chromosome. Tetraploid, that is the four sets of chromosome. And poly means many sets of chromosome. When the chromosome number is not the exact multiple of the haploid set, it is described as a aneuploidy. Aneuploidy is either addition or deletion of one or more chromosomes to the total number of chromosomes in a cell. So there is a hypoploidy and hyperploidy. This is a monosomy. This is a deletion 2n minus 1. Nullisomy, that is 2n minus 2. Hyperploidy addition, that is the 2 trisomy 2n plus 1, and tetrasomy, that is the 2n plus 2. Next, that is the structure of chromosome. So this is the ultrastructure of chromosome. It shows different structure, that is the pellicle. Pellicle is the outer covering of the chromosome. Next, that is the matrix, chromosomal ground substance having chromonomata. So this is the ground substance with chromonomata. Chromonomata, each chromatid consists of two coiled chromatin threads. It has number of chromomeres. So these are the number of chromomeres. Next, that is the primary constriction. It is also called as a centromere or kinetochore. So this is the primary constriction. It is also called as a centromere. The two chromatids are held together at a point. It is called as a centromere. Two chromatids are held together. It is a centromere. Next term, that is the secondary constriction. So this is the secondary constriction. It is also called as a nucleolar organization because it is responsible for the formation of nucleus. Next, that is a satellite. So this is the satellite, small spherical part of chromosome present distal to nuclear organizer, and that is the telomere. So it is a telomere, tip or terminal end of the chromosome. Next, types of chromosome. So according to the position of centromere, there are four types of chromosomes. First is metacentric. So it is a V shape. So chromosomes with two equal arms, centromere at the center. So these are the two equal arms and centromere at the center. So it is a V shape. It is called as a metacentric. Second type is a submetacentric. It is L-shaped chromosome with unequal arms. Centromere is located little away from the center. So this is the little away from the center. It is a L shape. Acrocentric, it is a J-shaped chromosome with one very short arm and one long arm, and centromere is located little away from the end of chromosome. One arm is very short, second arm is very long, centromere away from the end of the chromosome. And last, that is the telocentric. It is a rod shape. It is a rod shaped chromosome with only one arm, and centromere is located at the tip. So these are the different types of chromosome according to the position of centromere. That is the next, that is the difference between X and Y chromosomes. So this is the X chromosome, Y chromosome. X chromosome is longer, straight and contains more genetic material. Y chromosome is short, straight in man and contains less genetic material. X chromosome contains large amount of euchromatin and less amount of heterochromatin. On the other hand, Y chromosome contains small amount of euchromatin and more amount of heterochromatin. X chromosome is metacentric, Y chromosome is acrocentric. It is a X chromosome is present in male and female. Y chromosome is present only in male. Non-homologous region contains X-linked genes. In Y chromosome, non-homologous region contains Y-linked genes. X chromosome determines female sex in human, whereas Y chromosome determines male sex in human. What is meant by euchromatin? So it is the portion of chromosome stained lightly, non-condensed state. Euchromatin is genetically active and it is rich in DNA. On the other hand, heterochromatin is the portion of chromosome stained darkly, more condensed state. Heterochromatin is genetically inactive and rich in protein. X and Y chromosomes contains both homologous and non-homologous segments. Homologous segment contains similar genes, while non-homologous segment contains dissimilar genes. X and Y chromosome contains both homologous and non-homologous segments. Homologous segment contains similar genes, while non-homologous segment contains dissimilar genes. Next term is a linkage. So what is linkage? Linkage is a tendency of two or more genes to remain together in the same chromosome and inherit together. It is called as a linkage. So it is a tendency of two or more genes to remain together in the same chromosome and inherit together. Linkage groups. All genes which are located in single chromosome are called as a linkage groups. The number of linkage group in man are 23. Next, that is the types of linkage. Incomplete linkage and complete linkage. Incomplete linkage. The linked genes which are located on the chromosomes and separated during crossing over are called incompletely linked genes, and thereof inheritance is called as a incomplete linkage. Linked genes which are located on the chromosomes and are separated during crossing over are called incompletely linked genes, and inheritance called as a incomplete linkage. For example, genes present on homologous region of X chromosome shows incomplete linkage. Complete linkage. The linked genes which are closely located in chromosomes do not segregate and inherit together. They are called as completely linked genes, and mode of inheritance is called as a complete linkage. So in this, linked genes closely located do not segregate and do not inherit together. For example, genes present on non-homologous region of sex chromosome shows complete linkage. Sex linkage. The genes associated with sex chromosomes and inherit together through sex is called as a sex linkage. The genes associated with sex chromosome and inherit together through sex is called as a sex linkage. There are two types: incomplete sex linkage and complete sex linkage. In incomplete sex linkage, the genes which are linked with homologous region of X and Y chromosomes and do not inherit together are called incomplete sex linked genes, and their mode of inheritance is called as incomplete sex linkage. Its examples are total color blindness, retinitis pigmentosa, and nephritis. Complete sex linkage, the genes which are linked with non-homologous region of X and Y chromosomes and inherit together are called complete sex linked genes, and their mode of inheritance is called as complete sex linkage. Its examples are red-green color blindness, hemophilia, myopia, and muscular dystrophy. Crossing over. Crossing over is a process that produces new combinations, recombinations of genes by interchanging and exchanging of corresponding segments between non-sister chromatids of homologous chromosomes. Crossing over is a process that produces new combinations of genes by interchanging and exchanging corresponding segments between non-sister chromatids of homologous chromosomes. It occurs during pachytenoprophase first of meiosis. The term crossing over was coined by Morgan. The mechanism of crossing consists of four sequential steps, such as first is synopsis, second is tetrad formation, third is crossing over, and fourth is terminalization. The phenomenon of crossing over is universal and it is a necessary for the natural selection because it increases the chances of variation. So in this lecture, we have completed chromosomal theory of inheritance, chromosomes in that number of chromosomes, structure of chromosome, types of chromosome, sex chromosomes, linkage, types of linkage and crossing over. So today we will stop here. Thank you.